Good morning to all. This is my presentation on prophylactic action of flaxseed oil against cyclophosphamide induced oxidative stress in mice liver. Myself Dr. Brij Mohan Singh, Associate Professor, Department of Zoology, Sri Kalyan Government Girls PG College, Sikar, Rajasthan. Introduction. Recently, an interest has been generated to develop potential drug of plant origin for the modification of drug toxicity. Plant products appears to have an advantage over the synthetic compounds in terms of low or no toxicity at the effective dose with the no side effects. Keeping in the view the fact this study was carried out to evaluate the role of flaxseed oil against the cyclophosphamide induced oxidative stress in mice liver. Flaxseed oil is a major source of lignan. They are widely distributed in plant derived food items and are believed to protect against cancer. The richest source of lignan is flaxseed, which has been reported contain glycoside and secoisolericyresinol as a major lignan together with a small amount of metaresinol, isolericyresinol and pinoresinol. Prasad 2005 found that secoisolericyresinol isolated from the flaxseed reduces the hypercholesterolemic atherosclerosis, decrease serum cholesterol, LDL-C and lipid peroxidation products. This increase high density lipid C and antioxidant reserve. Flaxseed were found to inhibit human breast cancer and metastasis and reduce expression of insulin like growth factor and epidermal growth factor receptors. Manda and Bhatia evaluated a role of lignan rich flaxseed oil against the cyclophosphamide induced oxidative stress. Oxidative stress refers to the cytotoxic consequence of reactive oxygen byproduct, superoxide anion and hydroxyl radicals, which are generated as a metabolites of normal and aberrant metabolic processes that utilize the molecular oxygen. Oxidative stress lead to the lipid peroxidation, protein and carbohydrate oxidation and metabolic disorders. Therefore, the present studies aim to reveal the mechanism of hepatoprotection by the flaxseed oil in albino mice. Material and methods. Animal selection. 40 adults of 6 to 8 week Swiss albino mice from the inbred colony, body weight 150 to 200 gram were having selected for from Animal Care Center, Department of Zoology, University of Rajasthan, Jaipur. The animal were kept in a 12-hour light and dark cycle of temperature of 22 degrees centigrade and with the relative humidity of 50 plus minus 20%, was ventilated with the filtered non-cycled air. Experimental procedures were carried out according to the guideline of National Institute of Health and Animal Care. Different concentrations of flaxseed oil were given orally to the mice for the selection of optimum dose in terms of milliliter per kilogram body weight. After supplementation of 15 days, the sublethal concentration of pro-oxidant cyclophosphamide has been injected to the animal. The survivability and body weight were checked up for the 30 days. The dose of pro-oxidant cyclophosphamide, which produced 50% mortality with the 30 days, were estimated. The group which showed maximum survivability and healthier condition were selected for the study. Experiment. Swiss albino mice have been divided in 3 groups. Control group, which were received at not any other treatment. The second group was administered with the cyclophosphamide pro-oxidant. Third group was already cyclophosphamide injected and given a single dose of flaxseed oil orally. Observation. The mice were sacrificed at the various intervals and between the 1 to 30 days. Mice of all 3 groups after treatment were microscopically observed and survivability, body weight changes, sickness induced due to the pro-oxidant, serum markers, histopathological analysis by standard methods. Results and discussion. For the sake of convenience, this section was divided into 2 steps: assessment of lipid and assessment of protein markers. First, assessment of lipid. The low density of lipid LDL level of mice significantly decreased in the comparison to the control group in the CP administered group second, and the further increase in the third group, which is CP administered with the flaxseed oil. This could be due to the protective effect of lignan complex of flaxseed oil. Increase in HDL due to the flaxseed supplementation may be explained by the stimulation of lipid peroxidation. A considerable change in cholesterol activity, which increase cyclophosphamide administered group second and CP plus flaxseed supplementation group third. This elevation might be attributed to the protective effect of lignan complex. The difference of all figures and data were significantly determined. Comparison of triglyceride level control and experimental group flaxseed supplemented group third has revealed a significant elevation in the plasma triglyceride level. Elevated triglyceride level could be due to the decreased activity of lipoprotein lipase enzyme liver in acute myocardial ischemia. Lipid profile. Lipid profile alteration in the lipid profile in CP treated rat attributed to enhance the lipid biosynthesis by cardiac cyclic adenine monophosphate may be responsible for the triglyceride elevation. Assessment of serum profile. Extent of hepatic damage were assessed by the measuring the level of alkaline phosphatase, alkaline transaminase and aspartate transaminase in the circulation. The effect of flaxseed oil pretreatment on the serum TNF alpha, ALP, ALT, AST activities after cyclophosphamide administration were increased significantly. TNF alpha serum is a prominent element in the cytokine hypothesis of heart disease and has been focused on the research in myocardial ischemia. However, majority of clinical dietary intervention trials were investigated the effort of flaxseed on markers of hepatic inflammation. A single CP dose resulted in a significant increase in activity of ALT, AST and ALP in the liver of mice. However, pretreatment with flaxseed oil significantly lowered the serum level of hepatic markers. These data suggest a hepatoprotective role of flaxseed oil. This is the table showing the levels of cholesterol, triglyceride, HDL, LDL in liver and serum markers TNF alpha, ALP, ALT and AST in the plasma, control and experimental group of mice. All the data are represented in standard deviation plus minus 5. This is the LDL, HDL, cholesterol, triglyceride, TNF alpha, ALP and ALT and the group first and group second. These are the increment percentage and this in the second group and this is the increment percentage in the third group. This is the graph showing the various parameters LDL, HDL, cholesterol, triglyceride, TNF alpha, ALP, serum ALT and serum AST. This is group first and this group second CP exports and this is group third with CP and flaxseed oil combined ameliorating effect of these two substances on the mice liver, showing the comparison parameters cholesterol, triglyceride in the graph in the control and experimental groups. This is the photograph of original hepatocytic cells. This is the original hepatocytic cells in control group and these are the sequential deterioration of hepatocytes or liver cells in the A, B, C in the showing cytotoxic effect in cyclophosphamide treated in mice liver. Conclusion. In this study, flaxseed oil showed the dose dependent protective effect and reduced cyclophosphamide induced hepatotoxicity in mice. Administration of flaxseed oil to mice for limited days prior to cyclophosphamide injection attenuated to the serum level of hepatic markers. Hepatopathological examinations confirmed the cytoprotective effect of flaxseed oil and for improving defense mechanism in physiological system against oxidative stress and inflammation caused by cyclophosphamide. This oil is more useful against CP induced liver damage because CP induced tissue damage might be elevated by the antioxidant activity. Free radical scavenging, increased activity of antioxidant defense system and because the defense system and the membrane stabilizing property of flaxseed oil, because flaxseed oil is an additive therapy and herbal medicine for tumors. It could be a potential agent for the safe supplementation agent against the side effects of chemotherapy. Therefore, it can be concluded that the flaxseed oil is a very important supplementation in the antioxidative activity of cyclophosphamide. Thank you.